Labosys Bioreactor, Fermenter is a vessel that provides a controlled, aseptic environment, temperature, pH, aeration, agitation, for cultivating microorganisms, cells, or tissues to produce bio-based products like pharmaceuticals, vaccines, enzymes, and beverages. While sometimes used interchangeably, fermenters generally focus on industrial, high-oxygen demand microbial growth, whereas bioreactors constitute the broader category for cultivating diverse, often delicate, cell types. Key components and features. Vessel. Typically stainless steel, for industrial scale, or glass, for lab scale, often designed for sterilization in place, SIP, or autoclaving. Agitation, stirring. A central shaft with impellers, agitators, ensures proper mixing of nutrients and cells. Aeration, Sparger, introduces oxygen or other gases needed for aerobic processes. Control system. Monitors and adjusts critical parameters like temperature, pH, and dissolved oxygen to optimize yield. Types of bioreactors, fermenters. Continuous Stirred Tank Reactor, CSTR, the most common industrial type, using a motor-driven agitator for intensive mixing. Airlift Reactor. Uses air movement for mixing, reducing shear stress on cells, no moving parts. Bubble column. A simple cylindrical vessel with a sparger at the base. Photobioreactor. A transparent vessel using light to grow photosynthetic cells like algae. Packed bed, fluidized bed. Used when cells are immobilized on particles. Key differences between fermenter and bioreactor. While the terms are often used interchangeably, this article offers a useful distinction. Fermenter, typically specialized for microbial fermentation, bacteria, yeast, fungi, often requiring high agitation and aeration, e.g., antibiotic or beer production. Bioreactor, a broader, more versatile term often used for more delicate mammalian or plant cell cultures, where low shear, precise control is prioritized. Applications. Pharma, biotech, production of vaccines, monoclonal antibodies, and therapeutic proteins. Food and beverage, beer, wine, and yogurt production. Industrial biotech, enzymes, biofuels, ethanol, and organic acids. Environmental, wastewater treatment. Labosys fermenter bioreactors are sterilized, controlled vessels used to cultivate microorganisms, animal cells, or plant cells to produce valuable products. They manage parameters like temperature, pH, and oxygen to optimize microbial growth and metabolic output for applications including pharmaceutical production, antibiotics, vaccines, food processing, yogurt, beer, enzyme production, and wastewater treatment. Key uses of fermenter bioreactors. Pharmaceutical and therapeutic production. Used to create large quantities of recombinant proteins, insulin, vaccines, monoclonal antibodies, and antibiotics like penicillin. Food and beverage industry. Essential for producing fermented items such as yogurt, cheese, beer, wine, and vinegar. Industrial enzyme production. Cultivating microbes to produce enzymes like amylases, proteases, and lipases used in detergents, food processing, and textile manufacturing. Alternative protein generation. Utilized for growing single-cell proteins in modern applications like cultivated meat. Environmental technology. Used in wastewater treatment plants, specifically in activated sludge processes, anaerobic digestion, and membrane bioreactors to break down organic matter. Biofuels and biochemicals. Production of biofuels, ethanol, biodiesel, and industrial chemicals like organic acids. Operational context. Fermenter. Typically refers to systems optimized for high-density microbial processes, bacteria, yeast, fungi, often relying on strong agitation and aeration, aerobic. Bioreactor. A broader term often used for sensitive cultures, such as mammalian or plant cells, used in gene therapy or vaccine production, which require lower shear stress. Types. Common types include stirred tank, most popular for industrial use, airlift, and solid-state fermenters.